Hi, today we are going to discuss something about viruses. So basically, viruses are small non-cellular entities and they are having a molecule of RNA or DNA inside a protein coat. So viruses are basically smaller structures and they are smaller than the prokaryote. The size can range from 0.01 micrometer to zero point 0.4 micrometers. And the viruses are having the viruses multiply inside a living organism. They are non-cellular, but they multiply under a cellular organism. So they are also known as obligatory intracellular parasites. Viruses are having protein coat as well as nucleus, which is also termed as nucleocapsid. So this nucleocapsid, basically the capsid which is made up of protein, basically protects the viral genome from nucleases and other kind of enzymes. So the protein coat, that is capsid, it can be single shelled or can be double shelled. The main function of the capsid is to protect, and also it helps in recognition of the host cell. It helps in recognition of the host cell also beside protection. The infectious particle, which is basically infecting the host cell, is also termed as virion, and the function of the virion is to transport the DNA or RNA into a host cell. Um, this transport of DNA, RNA, why it is taking place? It is taking place because virus itself is unable to express its genome because it is lacking cellular machinery, it is lacking all the enzyme. That is why it is termed as non-cellular. So it needs a cellular host which is having proper machinery to replicate the genome as well as to express the genome. So express the genome into protein, from the DNA to RNA and from the RNA to protein, so that the genetic material get expressed. So it needs a host genome. That is why this virion is there, and virion function to transport the DNA or RNA into the host genome, so that the virus materials can get expressed, because host cell is the only cell which is having all the cellular machinery, all the important enzymes, all the important material that is needed to express the RNA or DNA of virus particle into protein. So the structure of virus basically vary diversely, and it is having a wide range of size, shape, and chemical components. The term, we know that nucleocapsid is there, and the protein coat is, that is capsid, is having single shelled, can be single shelled, it can be double shelled. And the some particles which are enveloped and some particles which are non-enveloped. So there are basically two types of virus on the basis of envelope. One is enveloped, and we can also say enveloped virus, and the other one is non-enveloped virus. And the enveloped virus sometimes have glycoproteins on their envelope, and these are also termed as spikes, or these are also termed as peplomeres. These helps in recognition of the host, and it act as receptors. So what happens, that the viral genome, which are smaller in size, also known as bacteriophage. The smaller genome is bacteriophage. The genome of a virus can consist of DNA, and if it is DNA and it is double stranded, or it can be single stranded, and the DNA can be circular, DNA can be linear. So there can be the genetic material. Genetic material can be single. Genetic material can be double-stranded, and it can be circular. It can be linear. So, if it is RNA and it is present as single stranded, it will be called as plus sense. And if it is present the other strand will be termed as minus sense. So if the plus sense RNA serve as messenger RNA, and the plus sense is in some regions are present, and in some region minus sense is present, then it will be termed as ambisense. So there are two types of genome. One is DNA, one is RNA. So we know that RNA is single stranded basically. So if there is single stranded RNA, then it can be linear or it can be circular. If the single stranded RNA is there, it can be having plus strand, and we can also say plus sense or minus sense. And plus sense RNA will serve as messenger RNA. It will code for messenger, it will serve as messenger RNA. It will help in coding of protein, while the minus sense will not be helping in messenger RNA, so or not be working as messenger RNA. But the but the genome which are having both the plus sense and minus sense are also known as ambisense. So, based on the organization of the viral genome, they can be divided into three categories. Monopartite, they can be called as segmented, and they can be also termed as multipartite. So, monopartite are those kind of genome which are not at all segmented. They are one segment which is entering into the virus particle, or which is having which is inside the virus particle. It is one segment which is contained by virus particle, while the multipartite can have more than one segments. They can have 10 to 12 segments. From 1 to 2 they can have 10 to 12 segments. So these particles, these segments are contained only inside one phage or one virus. So these kind of viruses are also known as multipartite, or these kind of genome are also known as multipartite genome. So rest about the virus. This was all about the structure of virus, and this was all about the inside of virus, that what is inside the virus. So the rest we can cover in next upcoming videos. So wait for it and kindly join. Thank you.